Welcome everyone. My name is Allison Gruber from Indiana University. I'm chair of the American College of Sports Medicine Biomechanics Interest Group, or ACSM BIG. I'm joined today by Max Paquette, one of the co-chairs of ACSM BIG. Our other co-chairs are Ross Miller from University of Maryland and Rumit Singh from Old Dominion University. This is our third installment of our webinar series. Our guests today are Becky Krupenevich, postdoc in the Department of Engineering at UNC Chapel Hill, and Billy Clark, PhD student in the Department of Engineering, also at UNC Chapel Hill.
Right. So as Ross and Rumit explained last time in the previous webinar, the whole point of this webinar is that we are able to bring content to our membership, not just at the annual meeting. Of course, this year we didn't have one. So with these webinars, it just gives a chance to have a bit more exposure for our members and give a number of biomechanists a chance to present their work. Before we get going, I wanted to thank all our sponsors that are around Allison and I's heads right now. Without all their help, it's tough to make anything happen really at the annual meeting and even to help support these types of webinars. So today, Becky and Billy will be discussing the use of deep neural networks for tracking ultrasound images of gastrocnemius muscle tendon junction. So without further delay, Becky, you could start.
And hopefully everyone can see that okay?
Yes.
Mm-hmm.
All right, thank you so much for the introduction. I want to start by giving a brief outline of what Billy and I are going to talk about today. We're going to break this talk up into two sections. I'm going to start by talking about using deep neural networks to automatically track the position of the gastrocnemius muscle tendon junction in the context of age-related changes in Achilles tendon properties. And Billy is going to talk about the development and use of dual probe ultrasound imaging techniques to investigate the interaction between muscle and tendon during functional activities and with changes due to aging. Billy and I both work in the Applied Biomechanics Lab in the Joint Department of Biomedical Engineering at UNC Chapel Hill and North Carolina State University. The primary mission of the UNC Applied Biomechanics Lab is to preserve independent mobility in our rapidly aging population through scientific, technological, and translational innovation. I'm currently a second-year postdoc in the lab, and a lot of my work is centered around understanding age-related changes in muscle-tendon interaction and how these changes affect mobility in older adults. So we know that older adults walk slower and with a higher metabolic cost than young adults. These functional impairments are associated with a loss of independence and quality of life and are thought to stem in part from a substantial reduction in mechanical power during the push-off phase of walking, as we can see in this figure by Franz and Kram, with older adults displaying smaller ankle contributions to trailing leg work compared to young adults. However, interventions designed to strengthen the plantar flexors have been generally unsuccessful at improving push-off power, suggesting that the plantar flexor muscles alone are probably not the only contributing factor to reduced push-off power during walking. In fact, mechanical power is not solely generated by muscle mechanical output, but instead arises from the interaction between the active triceps surae muscles and the passive Achilles tendon. Together, the Achilles tendon and the triceps surae muscles generate power through a catapult mechanism, where in the early stance phase of walking, the triceps surae fascicles generate force nearly isometrically, while the Achilles tendon is simultaneously stretched and stores energy. Then in late stance, the tendon rapidly shortens, which allows the triceps surae muscle fascicles to remain at a relatively slower shortening velocity, which promotes economical force generation and powers ankle push-off. But we don't get an effective catapult just by having an Achilles tendon. Musculoskeletal models and simulations suggest that optimal ankle function requires a tuned interaction between active muscle stiffness and passive Achilles tendon stiffness. For example, in the figures on the left, we see that decreasing Achilles tendon stiffness results in greater gastrocnemius activation and greater triceps surae metabolic energy expenditure. Now, this is important because we know that with age, Achilles tendon stiffness has been shown to decrease by up to about 40%, at least in humans, which may act to disrupt the tuned neuromechanical interaction with the triceps surae during walking. And as part of our ongoing research in the lab, we aim to understand the age-related effects of tendon stiffness on muscle and joint mechanics and metabolic energy costs in older adults. To measure tendon stiffness, we need two things. We need to be able to decompose ankle moment into tendon force, and we need to be able to measure tendon elongation. And now this is where things get a little bit tricky and challenging in measuring tendon elongation. Well, so how do we measure that? Well, one common way is to subtract ultrasound-based measures of longitudinal muscle length change from the overall length of the muscle tendon unit. But this type of indirect measurement frequently yields implausible outcomes. In some cases, outcomes that suggest passive tendons are somehow performing net positive work, which we know is not possible. So it's suggesting that there's some sort of fundamental problem with these indirect tendon estimates, either due to measurement inaccuracy or incorrect methodological assumptions. And given the growing use of ultrasound imaging to understand the functional role of tendons in dynamic movement, we really hope to promote methods that are more accurately capturing tendon behavior. The gold standard for reliably estimating tendon behavior and muscle-tendon interaction during functional activities is direct measurement of the position of the muscle-tendon junction, or MTJ, which is defined as the most distal insertion of the muscle into the free tendon, shown here in red. The problem is, manually labeling MTJ position in each frame of a dynamic ultrasound video is massively time-consuming. A 5-second video recorded at 70 frames per second may have 350 frames and is just not going to be practical for larger data sets or data sets that span multiple cohorts with many experimental manipulations or conditions. Additionally, this direct MTJ method requires a level of dynamic imaging expertise and knowledge of muscle tendon architecture that may be prohibitive for clinicians or other researchers. So what the field really needs is an automated method to accurately track muscle tendon junction position that is accessible to clinicians and rehab specialists. So in the next few minutes, I'm going to summarize our efforts to automatically track the gastrocnemius muscle tendon junction using open source deep learning techniques. Deep learning is a subfield of machine learning that uses neural networks to make up these different algorithms. And deep neural networks have become a really popular tool for image identification, particularly in the medical sciences. And that's because deep neural network-based architectures are made up of multiple hidden layers, or deep layers, that learn common features of the training images, such as shading or shapes, or in this example on the screen, edges or combinations of edges, and then use that knowledge to recognize those features in future images. So this technique may be an ideal solution for tracking features in ultrasound images. And to construct our networks, we used DeepLabCut, which is an open source software package that is easily accessible for people who don't have extensive training in machine learning. I want to mention here that a lot of this work was spearheaded by a really talented BME undergraduate student, Callum Funk, who joined the project early on and has really put a lot of work into this. And one of the first things that we wanted to do was evaluate the efficacy of these deep neural networks at accurately tracking the medial gastrocnemius MTJ position from ultrasound images collected during both controlled loading in isolated contractions and during physiological loading from treadmill walking. We also wanted to know how the networks would perform when given images from a dynamic condition that it was not trained on. So our networks were only trained using images recorded during walking. And then the images recorded during maximum isometric voluntary contractions were then used in the evaluation process. Finally, we wanted to replicate the small data sets that are common in biomechanics studies. So in total, we used data from just 15 young adult subjects. The first thing that we needed to do was label a bunch of images and create our training set. So one investigator manually labeled the gastrocnemius MTJ in images collected from each subject. And we call this the ground truth or the true position. In order to make sure that each subject was presented to the network as a novel subject, we actually created 5 networks, which are illustrated in the figure here, where each row represents one network. Each network was then trained on data from 12 training subjects, which are shown in white, and was evaluated on 3 test subjects, which are shown in black, that were not introduced to the network prior to the evaluation stage. So you can think of this as a test of how well the networks can generalize between subjects. Okay, so now that we've got our training and test set sorted, it's time to train the network. But first, we need to decide how many iterations and how many frames of data the networks needed during training in order to minimize error. In the next few slides, we're going to quantify error as the root mean square error and the mean absolute error, or MAE. So to determine training set size, we systematically varied the size of the training set and trained 45 distinct networks, or 5 at 8 different set sizes. And we found that RMSE and MAE, our error metrics, decreased with a greater number of frames and were both relatively stable from about 500 to 1,000 frames. We looked into that a little bit further, and we determined that a training set size of just 480 total frames, or just 40 frames per subject, was sufficient to achieve an overall low error value. And then to determine the number of training iterations, we first trained to a large number of iterations and took snapshots of training progress at several different time points. From that, we put together this figure where we can see that after a steep initial drop at about 3,000 training iterations, RMSE fell below 5 millimeters and remained stable as the number of iterations increased, and was, I guess, technically minimized at around 18,000 training iterations, but we just went ahead and decided to train to 24,000. So now that we've selected our parameters and we've trained our networks, we're ready to track a video. On the top, make sure it's playing. On the top here, you're seeing the tracked ultrasound video recorded during walking, and on the bottom, you're seeing the corresponding position of the tracked MTJ. Now, of course, I wanted to start by showing you a really good video, but they weren't all like that. So we did have some images that didn't track as well, and this is an example of one of them. It's a little bit hard to see at full speed, so I'm going to slow it down a bit during the noisy section at the end. And as you can see, there are some instances where the estimated position is not accurately tracking the position of the MTJ. And these poor tracking results may be due to a variety of different factors, such as narrow space between the deep and superficial aponeuroses, or you can think of that as just being like a really small medial gastrocnemius muscle width, or aponeuroses that don't fully converge. Did that video play? Okay. Or aponeuroses that don't fully converge, but with a small data set, it's really hard to expose the networks to the full range of MTJ characteristics that they might encounter in novel images. So we wanted to figure out a way to reduce some of this noise in videos that track okay, but have a few instances of poor tracking. For example, in this figure, we see the ground truth MTJ position plotted as the gray line. Estimated positions from the neural network are then shown as the green dots. Here, for simplicity, we're just showing every 4th point. And we can see that some of the estimated positions fall away from the true position, or that gray line. To reduce noise, we implemented a modified median filter on data points that fell below a confidence threshold of 98%. So the confidence score is illustrated in the bottom plot as the solid gray line, and the confidence threshold is illustrated by the dashed gray line. And the confidence score is a value from 0 to 1, and you can think of it as representing the neural network's confidence that the MTJ is visible in that frame. So any points that fell below the confidence interval, which are shown here in red, were replaced with the median of the 3 frames preceding and the 3 frames following the poorly tracked frame. So here in blue are the filtered frames, and you can see that in this case, the filter did a pretty good job of correcting the noisy data points, but points that were above the 98% confidence threshold were not affected. And this is one of the reasons that we chose this filter method. We wanted to decrease some of the noise on certain points, but otherwise we wanted to leave the rest alone. We wanted to just maintain our data's accuracy. So here we see filtered and unfiltered results for each individual during walking. And as a quick reminder, these data, the walking data, represent novel subject performance, or how well the network tracked when seeing a specific subject's image for the first time. The filtered data are shown in blue, and the unfiltered or raw data are shown in green. And we can see that 11 out of our 15 subjects tracked really well with an RMSE of less than 2 millimeters. In this next plot, we see individual subject results for the MVIC images. And here, I'll remind you that the networks were only trained on walking data. So these images actually evaluated the network's ability to generalize to both novel subject and novel condition images. I will note that we're missing a subject in this part of the analysis due to a data collection error. So on the bottom plot there, we've only got 14 subjects. There's no subject 1. But similar to the walking evaluation, we can see that the RMSD for most subjects, 11 out of 14, was below 5 millimeters. Finally, as one additional performance metric, we can look at the proportion of valid frames, which is the proportion of frames within a 5 millimeter threshold of the ground truth. In this figure, solid lines represent walking and dashed lines indicate isolated contractions. The red vertical line marks the 5 millimeter threshold. And we can see that on average, about 95% of tracked frames for walking and about 90% of tracked frames for the MBC data were classified as within the validity threshold. So how do our results stack up with others? Well, our network performance was comparable to, or in some cases a bit stronger than, other automated or semi-automated methods. The bars in green represent the RMSD from the current network. I think I have unfiltered and filtered both on there. And they're compared to results from a couple other studies in the literature. There are some limitations to this technique that I want to note. First, the networks we describe here are only appropriate for tracking videos in which the MTJ is visible in all frames, because we don't have the capability of extrapolating position information when the MTJ is not in view. Also, the network that we used, which I didn't touch too much on, MobileNet V2, it's a shallower network compared to some other network architectures out there. And so it may not be suitable for large images without downscaling, but it does come with some benefits. This network allows for faster training and analysis and can be run without a high-end GPU, which makes it ideal for researchers with limited computing resources. The faster processing speed of MobileNet also makes it ideal for future studies that aim to track in real time. Despite these limitations, we think that our results provide support for the use of open source software for creating deep neural networks to reliably track MTJ positions in ultrasound images. Just sort of bring this all back around to the beginning of the talk, I personally will be using this tool to investigate age-related changes in Achilles tendon stiffness and how those changes may affect mobility in older adults. But beyond immediate uses for our work on changes in tendon properties and aging, we think that this approach to MTJ position tracking is an accessible and time-saving solution and has broad applications for a lot of different fields like rehabilitation or clinical diagnostics, for example, as a real-time biofeedback paradigm to target specific muscle tendon behavior. And it's also accessible to people who don't have expertise in ultrasound imaging or deep learning. And I do want to share that once our paper gets through peer review, we do plan to make these trained networks publicly available. So in theory, somebody should be able to just take this network, plug in their ultrasound images and have some tracked data at the end of it. So that's really the goal here, is just making it accessible to more people who want to do this type of ultrasound image analysis. So I'd like to quickly acknowledge our funding sources and the support of the UNC Research Computing Group and the supercomputing resources at UNC. And now I'm going to pass it over to Billy, who's going to take over the next segment and talk about some of the ways our lab has used ultrasound imaging and research over the past several years.
Awesome. Thank you, Becky.
Oh, you're muted, Billy.
That always helps. All set? All right, so as Becky mentioned, I'm going to be talking about some of the ways that our lab has used ultrasound imaging in our research efforts. And the main focus of this portion of the talk is going to be on our dual probe imaging approach, where that allows us to examine muscle fascicle behavior and tendon tissue behavior at the same time. So we've applied this technique during isolated contractions and then also during walking in both young and older adults to try to uncover the musculoskeletal mechanisms underlying age-related mobility impairment. So first, I'm going to walk through some of the architecture of the structures that we generally image in our lab. And I think understanding the architecture is really key to understanding the ultrasound imaging techniques themselves. So one of the major units influencing walking performance is the triceps surae muscle Achilles tendon unit. And as many of the people probably watching this know, the triceps surae muscles, otherwise known as your calf, transmit substantial forces through the Achilles tendon, which generates a plantar flexor moment about the ankle. And we know that this plantar flexor ankle moment is the largest contributor to forward propulsion and provides support for the trunk. It's really important for regulating walking speed. And it's also significantly reduced by aging, as well as several pathologies like stroke and cerebral palsy. And there are three muscles that make up your triceps surae. There are two biarticular gastrocnemius muscles, the medial and lateral head, and these contribute to ankle plantar flexion and also knee flexion, and they make up about 43% of total triceps surae volume. And the remaining volume is from the one uniarticular soleus muscle, which is deeper and wider than the gastrocnemius muscles and contributes solely to ankle plantar flexion. And as I mentioned, these three muscles transmit their force through the Achilles tendon, which is actually made up of three twisting bundles of tendon fascicles known as subtendons. And these three subtendons arise from each muscle of the triceps surae. So you have two gastrocnemius subtendons, the medial and lateral gastrocnemius subtendons, and then one soleus subtendon. And our lab uses dynamic ultrasound imaging to characterize the tissue behavior of these subtendons. And we found that the gastrocnemius and soleus subtendons exhibit different displacement patterns during walking. So here's an ultrasound video showing the displacement of subtendon tissue. And the blue box is roughly surrounding medial gastrocnemius subtendon tissue, and the red box is roughly surrounding soleus subtendon tissue. And what we can do is we can track the displacement of these green nodes over time using a custom speckle tracking technique that I'll mention in more detail in a few slides. But if we stop the video during the stance phase of walking, we can see that the medial gastrocnemius subtendon tissue, has here indicated as blue dots, undergoes at least a different displacement profile than soleus subtendon tissue, here indicated as red dots. And we commonly refer to these differences in displacement as tendon tissue non-uniformity. So non-uniformity simply means significant differences between gastrocnemius subtendon behavior and soleus subtendon behavior. So more non-uniformity would mean greater differences. And we think that this non-uniform behavior of the Achilles tendon is really interesting because it could allow the individual triceps surae muscles to independently actuate. And this is important because the gastrocnemius and soleus do different things during walking. In particular, they contribute differently towards vertical support and forward propulsion. So maybe the non-uniform behavior of the Achilles tendon is allowing different contractile behaviors between the gastrocnemius and soleus muscles. But we also think there's a potential translational implication to tendon non-uniformity. So in older adults, we actually see a significant reduction in tendon non-uniformity during walking. So in young adults, non-uniform tendon tissue displacements during walking, and in older adults, uniform tendon tissue displacements during walking. And some of our prior work from our research group shows that as young adults walk faster, the level of tendon non-uniformity increases. However, in older adults, we see no such change. So some studies have looked at rat and equine tendons and have shown that older tendons have a reduced capacity for sliding between subtendons due to interfascical adhesions. And these adhesions may couple the individual subtendons and cause them to behave more as one aggregate tendon. And this could potentially restrict the independent actuation of the gastroc and soleus and potentially negatively impact forward propulsion and trunk support. And supporting this overall idea, the level of tendon non-uniformity positively correlates with peak ankle moment during walking. So overall, this alludes to some muscle tendon related mechanism, but in these experiments, we actually don't really know what's happening at the level of the muscle. And that's really the central idea of this line of research. We believe that the interaction between muscle behavior and subtendon behavior influences walking ability and is uniquely compromised with aging. And that central idea can really only be fully explored by employing different imaging techniques. So using ultrasound imaging, we have tried to address a couple main questions. So first, no empirical studies have really characterized the origins of non-uniform displacement patterns in the human Achilles tendon. So during a series of isolated contractions, we set out to characterize these origins and quantify the effects of aging on the role of muscle contractile dynamics in governing Achilles tendon tissue displacements. And then second, those previously observed correlations between more uniform Achilles tendon tissue displacements and reduced push-off intensity in older adults alludes to some muscle tendon related mechanism. So during walking, we set out to determine the functional implications from age-related changes to this interaction. But really there is a technological challenge here. For us to understand how muscle and tendon interact, we must simultaneously measure both muscle and tendon behavior. As Becky mentioned, we learned from past studies that when you indirectly estimate tendon behavior using muscle data, or vice versa, you often get inaccurate or even implausible results. So to investigate this interaction between muscle and tendon, like I mentioned, we implemented a dual probe ultrasound imaging approach where we placed one ultrasound probe over the calf muscle and then a second ultrasound probe over the Achilles tendon. So for the muscle, we place an ultrasound probe over the mid-belly of the medial gastrocnemius, and that allows us to simultaneously view the soleus in the same imaging plane. And then in post-process, we can estimate muscle fascicle behavior using an affine extension to an optic flow algorithm. We used UltraTrack. And this gives us estimates of fascicle length and pennation angle. And then we and others are also exploring using deep learning to more reliably track this muscle fascicle behavior. But then for the tendon, we used a custom two-dimensional speckle tracking technique that tracks the localized displacement of subtendinous tissue. So here during an initial frame of an unloaded tendon, we defined a grid of nodes along the length of the tendon. And then each node is surrounded by a kernel, which contains upsampled radio frequency data. And then for every subsequent frame, a search window updates the position of each node based on the location of radio frequency data that maximizes two-dimensional cross correlation. And then for each subtendon, we can average the displacement of the nodes defined within that subtendon. Although it is important to note that this technique does not account for very much intersubject variability of subtendon width. So we are exploring new techniques to define subtendon boundary conditions, namely through the use of 7T MRI. So we first set out to apply this dual probe imaging approach in young adults, performing a series of ramped maximum voluntary isometric contractions at several different ankle angles spanning a neutral 0-degree angle to a 30-degree plantar flexed angle. And our subjects sat in a Biodex dynamometer. And here our goal was to determine how muscle and tendon behave during a very controlled environment and sort of try to establish the origins of Achilles tendon non-uniformity. So first I'll show the average behavior at the tendon level. So here we see soleus subtendon tissue displacements in red and medial gastrocnemius subtendon tissue displacements in blue, shown from rest to maximum muscle activation back down to rest. And on average, what we found is that the soleus subtendon displaced about 51% more than the medial gastrocnemius subtendon in response to ankle moment generation. So essentially what we observed here was non-uniform tendon behavior in these young adults. Now, if we look proximal to that, we see anatomically consistent behavior at the muscle level. So very similar to the tendon, the soleus muscle shortened on average 76% more than the medial gastrocnemius. And this anatomically consistent behavior played out fairly well in the correlations, where here more non-uniform behavior at the level of the tendon on the y-axis positively correlated with larger differences in peak shortening at the muscle level on the x-axis. And our main interpretation of these data is that the triceps Surae muscle dynamics themselves can precipitate characteristic non-uniform displacement patterns in the human Achilles tendon. So next we set out how to see how this interaction between muscle and tendon changes with age. So here again, I show that same plot as earlier, the young adult subtendon behavior. Now, if you take the difference between peak gastrocnemius subtendon tissue behavior here and peak soleus subtendon tissue displacement here, what we called non-uniformity, and we plot those differences for each ankle angle, it looks something like this, where each dot represents the difference between those two peaks for each ankle angle. Now, if we do the same exact thing, but in older adults, we reveal that older adult tendons are 63% more uniform than young adults, despite undergoing significantly more displacement. So these findings may reflect interfascical adhesions in these older adult tendons. Now, if we bump up to the muscle level, we again see more uniform behavior in older adults. So the differences between gastrocnemius and soleus muscle shortening decreased by about 61% in these older adults. So overall, age-related tendon level differences were mirrored by muscle level differences. So during isolated contractions, we revealed more uniform subtendon behavior in older adults, a finding that may reflect interfascical adhesions and thus a loss of independent actuation between the gastrocnemius and soleus muscles. And we sort of think of this as a tendon up theory, where aging affects tendon behavior, which then negatively affects muscle behavior. But we also can't exclude a potential alternative interpretation, more of a muscle down theory, that aging may affect triceps Surae muscle contractile behavior, therefore yielding more uniform Achilles subtendon displacements. And this could be achieved through changes in protein biology, muscle coordination, perhaps a loss of viable motor units, or maybe the older muscles simply aren't generating enough force to elicit that non-uniformity. So far, we don't really have a clear picture to which explanation is more plausible, and we are currently using electrical stimulation to try to tease that out. But the question remains, how do these changes in muscle and tendon behavior affect walking performance? So to test this, we systematically altered the mechanical demand for forward propulsion by changing speed and then also using horizontal aiding and impeding forces. So we had subjects come in and walk on a treadmill for 1 minute at a range of walking speeds, and then again at 1.2 meters per second with a 5% body weight aiding force condition that was designed to decrease mechanical demand for forward propulsion. It simply pulled forward on you. And then a 5% body weight impeding force condition that pulled back on you and was designed to increase the mechanical demand for forward propulsion. And throughout, we collected dual probe ultrasound imaging and also motion capture. And the video looks something like this, where we have dual probe ultrasound imaging while the subject is walking on an instrumented treadmill. So on to results for this walking study. We actually did not observe a significant effect of age on peak ankle moment generation during walking. And we think this is likely because our subjects were walking at relatively slow speeds. However, 1.2 meters per second was actually the maximum speed where we could collect tendon ultrasound data. On the other hand, despite walking with identical experimental manipulations, our older adults walked with a reduction in positive ankle power and also walked with a reduction in positive ankle push-off work. So overall, these older adults exhibited hallmark deficits in push-off intensity. So our next question is, what happened at the muscle and tendon level? So using ultrasound imaging, we established the magnitude of Achilles tendon non-uniformity during the push-off phase of walking. So here I'm showing that result with young adults in black and older adults in gray. And the first thing I'll mention, similar to our findings during isolated contractions, is that older adults had more uniform Achilles tendon tissue displacements during walking. And these age-related differences in non-uniformity increased with increasing speed, where older adults were about 53% more uniform at the fastest speed, 1.2 meters per second. Second, we add that these age-related differences are even larger in response to impeding forces that were designed to increase mechanical demand for forward propulsion. And these were up to about 63% during that impeding force condition. So what do we find at the muscle level? Well, again, consistent with our previous results during isolated contractions, we revealed that older adults walked with smaller differences in gastrocnemius versus soleus muscle length change behavior. And next, we wanted to examine that relation between muscle and tendon behavior. So here we show tendon non-uniformity on the y-axis and muscle level differences on the x-axis. And with changes in speed, we actually didn't observe a significant positive correlation between muscle and tendon level behavior for young or older adults. And this might be due to changes in speed simultaneously altering the demand for forward propulsion and also vertical support. However, when we only alter the mechanical demand for forward propulsion using those horizontal forces, we reveal our first key finding. And that's during walking in young adults, across changes in horizontal force, the magnitude of Achilles tendon non-uniformity positively correlated with larger differences in muscle length change behavior. And interestingly, these correlations were absent in older adults. And they were accompanied by significant age by speed and age by horizontal force interaction effects on tendon non-uniformity and muscle level differences. And these suggest that older adult muscle and tendon level behavior were significantly less sensitive to changes in mechanical demand. So in combination with our previous results during isolated contractions, we interpret our cumulative findings to suggest that the Achilles tendon facilitates independent triceps surae muscle actuation in young adults, but restricts that actuation in older adults. And as we show in the next slide, this may have some functional consequences. So in young adults, the magnitude of gastrocnemius for soleus muscle length change behavior positively correlated with each measure of push-off intensity. So we have peak ankle moment, peak ankle power, and positive push-off work during stance. And in our view, these findings provide a muscle level determinant for previously observed correlations between Achilles tendon tissue non-uniformity and push-off intensity. And consistent with our overarching hypothesis that more uniform Achilles tendon tissue displacements disrupts triceps surae contractile behavior and thus ankle joint mechanical output, we actually did not observe any significant positive correlations between muscle level behavior and push-off intensity in older adults. So overall, we interpret our findings to suggest that the capacity for sliding between adjacent subtendons facilitates independent gastrocnemius for soleus muscle actuation in young adults, but restricts that actuation in older adults. And the resultant disruption in triceps surae muscle contractile behavior likely contributes to hallmark reductions in push-off intensity during walking in older adults. However, it is important to note that the majority of these conclusions are based on correlations that cannot themselves definitively convey causal links. With that, I'd like to thank our funding sources and collaborators on these projects, and I'm going to kick it back to Becky to close us out.
Yeah, so I'm just going to pop back in here and sort of sum all of this up. Billy gave a really nice summary of his work that showcases some of the ways that ultrasound imaging has been used in our lab's research in the past several years. Now, although the techniques that I talked about were specific to determining MTJ position in ultrasound images with practical applications for calculating tendon elongation and stiffness, we think that deep neural networks are becoming more and more popular and may soon have more use in biomechanics and ultrasound image analysis. We want to thank you for inviting us today, and we're happy to take any questions.
Awesome. Thanks, guys. Allison, you want to start things off?
Sure. Thank you for the great talks. We really appreciate it. So one question I have with the availability of some of these software programs that can help people that may not have a lot of experience with neural networks really get started in using them. What recommendations do you have to avoid some of the black box problems that can occur when software is kind of built for us?
Yeah, so I mean, I guess my recommendation would be just to read as much as you can about the techniques that you're using, instead of just pulling down, the open source software and just starting to sort of plug and play, really understand what you're doing and the parameters that you're using. We spent a lot of time over the summer playing around with the different parameters. I mentioned the iterations, the number of frames, but there were several others that we played around with as well, just to really sort of understand what we were doing. So we didn't feel like we were using this tool and, just using it to say we were using it, that we really understood what we were doing.
Great. Max.
Awesome. So, yeah, the work you guys have been doing over there at North Carolina with Dr. Franz has been really, really neat. And what you've presented just now is really kind of almost launches a whole new set of questions and ideas. And that's obviously that's the point, right? Sort of thing. With all this work right now, so especially just to kind of start off with you, Billy, showing what's happening between young and older adults in terms of the non-uniformity in tendons in the Achilles tendon. What's the next step in terms of, this is possibly related to mechanical output, which we know is important in terms of gait speed and whatnot and function. What's the next step then in terms of doing something about it? How do we, what you're observing at the tendon level, is that something that can be changed? How does that work? What does it look like for an applied scientist, so on and so forth?
Yeah, that's a great question. I think our first step is to inform assistive devices using these imaging techniques, where we know in older adults we see changes in tendon compliance, and perhaps through passive elastic exoskeletons you could augment those changes. I think also there's a lot of work in sort of the multi-scale research of muscle and tendon, where there is some evidence for the use of whether it be hyaluronic acid or other techniques where you could sort of change or augment any changes you see at the tendon level, or more of the cellular level. I think those are new and exciting research pathways.
Do we know anything about how training exposures, whether that might be strengthening or plyometrics or more dynamic tasks like sprinting or running or jumping, what these types of exposures might do to these tendon characteristics?
So I've mostly been only focused on age, so I don't want to speak out of line on that. I can note that the majority of my older adults are very active, and we still see these changes at the tendon level with more uniform subtendon displacements. So all I can speak on is anecdotally on that, but I'm sure there's a lot of literature out there that can speak to changes in cross-sectional area and in the tendon, changes in stiffness, changes in moment arm,
right.
all influence mechanical output of these structures.
Right. So this is new. The methods you're using are fairly new, I would say, at least for the general biomechanics sort of community, at least in the U.S. And so I wonder then, because you mentioned we know what training does to tendon, cross-sectional area, stiffness, so on and so forth. But specifically with regards to non-uniformity, I wonder if that's something we're missing, that, perhaps training is doing something else that is good or bad. It increases surface or cross-sectional area perhaps, but it might change this non-uniformity or some other type of effect that we don't know. And so I wonder if that might be something else to study with training studies moving on.
Yeah, another thing that is at least common in comparative literature is looking at changes in fiber type of the muscles and how they're affected. And with age, you get changes in the physiological cross-sectional area of type 1 fibers and type 2 fibers that may preferentially affect one muscle versus the other. And that's also affecting the entire drive down and force generation through the Achilles that could really complicate this.
Sure. Right. That's interesting.
Kind of to jump on that, Becky, you mentioned some potential applications of ultrasound work with feedback, visual feedback. Could you speak to what some of those studies might look like?
Yeah, so they're all hypothetical right now. I don't know of any studies currently using ultrasound as biofeedback, but it might be something along the lines of fascicle length change, or using MTJ to help understand perturbation behaviors in older adults, or just having them target specific muscle tendon behaviors during these biofeedback tasks. So you could display it on a screen and show them a target line, because I don't know that them viewing an image would really do much for them, but just really targeting these specific behaviors in a biofeedback sense.
Okay.
So I'm a little bit fascinated and curious with using diagnostic ultrasound in those dynamic tasks. Many people may not know how challenging it is to secure a probe over the skin of a moving human being, especially of the lower limb. And so this is always the question I have is, like, first of all, what are you guys doing in North Carolina that you can make this work so well? It just blows my mind. It's super tough tape of some sort or some device that you're using, but it seems to be working really well. And I think many might have questions about, what it is that makes this, feasible really in those dynamic situations.
And to jump onto that, do you seem to notice that it affects some people's walking gait more than others, older adults more so than young, or does it really depend on the individual?
Yeah, so we do use a lot of Coban. We do have a probe holder. So the specific probe that we use for imaging the fascicles and the MTJ, it's a longer low profile probe. And so that makes it a little bit easier. We have a 3D printed probe holder that kind of keeps it where we want it to be on the leg. And then we just Coban, a lot of Coban around it. And in terms of how participants walk with it on, I think that in biomechanics in general, we generally load up our participants with all kinds of equipment, and then we say, okay, now I want you to walk normally. But so I think it's just kind of that, we give them a long acclimation period on the treadmill, trying to get them used to walking with it on. And I think after a little while, it becomes just another thing that they're walking with. Billy, have you noticed many differences in, certain groups of populations?
The tendon probe can be a little more tricky because it's sort of in an uncomfortable spot. We use a custom orthotic to house that probe. And then we also wrap it in Coban. And we also use a sort of modified gel pad to create a little more comfort and also help with the image itself. And that helps. We do see probably a 3-degree range of motion at the ankle decrease, especially as you increase walking speed. And we can't really go much higher. I mentioned it, I think, but we can't go much higher than 1.2 meters per second for a couple different reasons. But one is impact. When you have heel strike, we actually lose the image quite dramatically, and that hurts our tracking schema. But the housing works pretty well, especially at speeds at or slower than 1.2 meters per second.
So that answers my other question. Selfishly curious about running. And I'm sure Allison is curious about that as well. It sounds like that's at this time, you're unable to do that with at least faster running. I would guess very, very, very slow running might be possible, but still then if 1.2 was a struggle, I'm sure running would be quite impossible.
1.2 is a struggle for the tendon probe. I think we could probably.
Oh, right. Got it. Yeah. Okay. Yeah.
Muscle probe would be much more feasible, I think.
Because you can sort of, how much compression, are you comfortable with when you're wrapping the probe on over the muscle? Because at some point that might change things and how you measure it, obviously.
Right. Yeah. The amount of compression that you're using when you're wrapping the Coban, it is something that you have to play around with a little bit. You don't want to compress the probe so tightly that you're actually, compressing the tissue underneath and changing the image and not allowing, the muscle, especially with the MTJ. If you think about the calf muscle and people with, bigger calf muscles, it sort of bulges out when the muscle contracts. So that can be really uncomfortable for the participant if it's wrapped too tightly, or it can sort of change the way things are moving underneath. So there is sort of a fine line, a balance to be found with wrapping the probes on the calf, for sure.
Yeah, and to add to Becky's point, I think it definitely became much easier when we started using the 3D printed orthotics that housed that ultrasound probe, because it sort of lowers the pressure on the leg.
Billy, do you have a photo of that? I'm not sure if you have I saw something in your presentation, but I'm not sure how clear it might have been for some that were not thinking about that at the time. But if you have something kind of zoomed in image of what it is, of what it looks like, or.
Becky, do you know if you have a photo of the muscle probe holder? I know I have a photo of the tendon probe holder.
I don't think that I do. I was actually looking for one yesterday.
Top secret. So,
if anybody's curious about it, we've shared it with a lot of labs. So.
Yeah.
It's one of those things that's always under so many layers of Coban when we're actually taking pictures during data collection.
Right. I gotcha. Well, no worries if you don't have one handy. See, Billy, are you still searching or?
I was, and I can share the tendon probe holder
Yeah,
again. But,
please, if you don't mind. Yeah, that'd be great. Right, I gotcha.
So this is the tendon probe holder, and we have to do that because it's a much the actual transducer sticks out, whereas like Becky mentioned, the muscle probe is longitudinal and is much more low profile.
Right. Got it. Yep. And so, how much does that weigh roughly?
It is made out of foam. It's very lightweight.
Okay, got it.
Yeah, this part is rigid here, and then we have rigid attachments for the markers, but everything else is just layered foam.
Very cool. Awesome.
And then for that muscle probe, we make it quite malleable for the actual thing that sticks over the ultrasound transducer, and then we have a hard housing that goes on top of that where we have markers.
Gotcha.
And that helps for comfort.
And that's just custom made, you know, that is 3D printed, these types of housing pieces.
Mm-hmm.
I had another question about the neural networks. I would imagine that even without these neural networks, just the labeling and the tracking of all of these images has a lot of inter-rater or investigator variability between people. So do these neural networks help to mitigate some of those differences, or are they only as good as the investigator doing the tracking?
Yes, so a little bit of both. You know, it'll certainly help with inter-rater reliability, but you know, it's at the end of the day, a human did label all of the training images, so it's only as good as that person who was labeling the training images. For our networks, it was the same person across all of the images, and we did that purposefully so that we didn't encounter that inter-rater error problem that you were talking about. But yeah, the goal of the networks, I think, is to try to make it a little bit more objective. But yeah, it is based on the human labels.
So if I were to start using the software, never having done ultrasound imaging and tracking before, so if I were to take the software and upload some images and try to get the tracking out for me, where is that, I guess, accuracy coming from? Is it coming from some large database of images that the neural network kind of conglomerated to find some training, or is it coming from, say, just the data from your one paper, for example?
Yes, so if you were taking, you know, the networks that we're making available, then it would be just coming from those data that we trained the network on. Now we are planning to obviously update with more than just 15 subjects. That was more of a feasibility thing. You know, can we make this work with a really small data set? But obviously the more images and the more subjects that we train the network with, the more accurate and the more able it will be to track those uncharacteristic MTJ features that I was talking about.
So are there any plans for maybe like a data sharing to kind of build a network of images that the neural network can then learn more from? Or are you more concerned about that inter-rater variability, again, kind of causing some issues with the more global database?
Yeah, you know, I think that there is an opportunity for sort of a data sharing situation where people just contribute their ultrasound images, their tracked ultrasound images, and it can always be the sort of thing where we go back and retrack them. But yeah, having this huge database or bank of images that we can train a network on certainly would not be a bad thing. I think that it would be a great opportunity in biomechanics and something to really push the field forward with these faster and more accessible data processing techniques.
So last question, still related though. So your neural networks seem to be trained on just about 400 something frames. So is there, how much better is it going to get with more subjects, more images, kind of this data sharing aspect of it only really needed 400 or so?
Sure, yeah, so that's a great point. With just 480 images, 40 from each of our subjects in the training set, we were able to get really low error values, and more so than, you know, some of the other automated or semi-automated tracking procedures in the literature. And so we were really happy about that. But one of the things that we did note as a problem, you could see that some subjects had really high, not really high, but higher error rates. And that's because the trained network didn't have that type of MTJ. It didn't know how to look for those characteristics. So in certain instances, we can certainly make it better. I don't know if I can give you a specific number as to how much more accurate, but I think that we can lower that number of subjects who had outlier data for sure.
Yeah. Okay. Yeah, that was what I was going to mention. So you think more data to train the network would eventually allow the 2 subjects that you removed from the analysis in your paper to eventually be included? Is that essentially?
Yeah, that would be the goal for sure. And I think that we could get there. We just need to have more examples of those specific types of, MTJ images to train the network on. And I think that might be including other tasks. So here we only trained on walking, but then we also wanted to test it on the isolated contractions. So if we train using all of that data, it might also sort of help to beef up the accuracy of the network.
Great, thank you.
So I guess I'm not quite sure, Allison, how much time we have or what we want to take it, but I just have one more question to ask you, and it's not really related to the science or the data, but more sort of about your journeys where you are now. Obviously, Becky and Billy, you're both very successful in biomechanics already, and you're moving through the academic ranks, PhD, postdocs, and Billy's moving on now for a postdoc as well. And so what types of recommendations or tips would you have to sort of those younger ACSM BIG members that are watching, and just sort of piece of advice as they move through the ranks, as they say?
Yeah, so I think the biggest thing that I would say is really do work on the networking piece. That's so much easier to say than to do right now in the middle of COVID when there are no in-person conferences. But there is Twitter, which I feel like I didn't really jump onto until a few years ago. It started being really active with it. But getting people to know your name and just chatting with people, when you can at conferences or on Twitter or wherever it might be. I think that really helps when you're looking for that next position, if it's the postdoc and, people know your name and kind of know what you do. And it just makes things a lot easier, I think, for your journey.
Makes sense.
Yeah, I'd say a similar thing. Really lean on your mentors. I think they're all so willing to help and provide so much valuable expertise that you should commonly seek them out and commonly have them, like Becky mentioned, know your name and really just lean on your mentors as much as you can and pave your own way, but rely on that help as well.
Awesome. I agree. Great advice. Cool. All right. Well, I guess just before we log off, I want to thank our awesome sponsors again around our heads, and hopefully we'll get to see you all in the late spring or early summer at ACSM big meeting if we get to that point in 2021. Thanks so much.
Great. Thank you.
Thanks for inviting us.
Yep. See you guys.
Thanks for coming.